Today, so less people have joined. 9 people. Hello everyone, is the screen visible and am I audible?
Yes, madam.
Yes, ma'am.
Yes, ma'am.
Good afternoon.
Okay, good afternoon. So
Good
let's
afternoon, ma'am.
begin with good afternoon. Let's begin with today's session. So welcome everyone to the NPTEL course on novel technologies for food processing and shelf life extension. And the course instructor is Professor Achin Mishra, sir, and he is in the Department of Agriculture and Food Engineering, IIT Kharagpur. I am Shikha Sharma. I am a research scholar in the Department of Chemical Engineering, IIT Guwahati. So today we have our live session regarding week 6 topics, which includes aseptic processing, hurdle technology concepts, natural antimicrobials and bacteriocines, and last is the lipids. I hope you have gone through the 4 lecture videos regarding these concepts, which we will be covering today in our live interaction session. So let's begin with the first practice problem. Also, if you have any doubt, so you can ask in between and at the end of the lecture. So this is our first conceptual question about aseptic processing. So which of the following statements they are accurate about the design of aseptic processing system? Statement 1 is saying aseptic processing design for commercial sterility relies on established principles of thermal bacteriology. Second statement is saying for optimal quality, heating and cooling should be conducted slowly as possible depending on the product's characteristics. Third is typically UHT process, which is ultra high temperature process, lasts for minutes. Thus, precise control of the residual time is crucial to prevent underprocessing. And fourth statement is saying that pre-sterilization of the product often involves heating. Pre-sterilization of a product often involves heating it to the required UHT temperature, then holding it at temperature for a specific duration of time to achieve desired sterility level, then followed by cooling. So anyone would like to give opinion on this?
Option B, madam. Two and third statement.
2 and
Three. Option B.
okay,
Op-
option B.
Option
Okay,
B, ma'am. 1 and 4.
Option B, ma'am.
1 and 4.
Option D, ma'am. 1 and 4.
So we are talking here about accurate statements. So answer is D, option 1 and 4, not option B. let's understand why. So first, let's understand what is aseptic processing. In the aseptic processing, there is a sterilization of food happens and also the sterilization of the package, and then these like the sterilized product is it is being put in the sterilized packaging material in the sterilized environment to create a safe, ready to eat, self stable. Like if we are even keeping it at the room temperature, shelf stable means if we are keeping at the room temperature, the product should be stable. That can be transported usually without even refrigeration. In aseptic processing, the design to achieve commercial stability is based on the well-founded principle of thermal bacteriology. So we know that here is it is our food. Food is coming. It is in flow thermal processing is happening, and it means it is a sterilization process. And then at the another side simultaneously, the package is also being sterilized, and the this food is being filled up in the sterilized package, and then final product we got, and this is the packing is also under sterilized condition, and we get our final product. So what is happening, as statement 1 is saying, aseptic processing design for commercial sterility relies on established principle of thermal bacteriology. So this statement, it is same as is no, it is not, it is same as the this statement, right? Which means it is correct, right? In aseptic processing, what is happening, the design we make for achieving the commercial stability so that all the bacteria will kill using the thermal treatment. So the statement 1 is true. Now let's see about statement 4th, so which is also true. So pre-sterilization of a product usually consists of heating the product to the desired UHT temperature. So we what we do is till desired UHT temperature we heat the product, maintaining this temperature for a some time. Like UHT treatment mainly occurs above 135 degrees Celsius, and it holds like around 135 for some few seconds. So for few seconds we do UHT treatment so that we achieve the desired degree of sterility. Then we subsequently cool the product, and we use the product and we store the product at ambient condition. Also at the like we filling also we do at the ambient condition or sometimes at the elevated condition, so that we can get the right viscosity of the product, and the finally the storage will be at ambient condition. So this statement, the statement 4th is also correct, right? Now option that statement 2 and 3, these are incorrect. So why these are incorrect? First is we can see here it is saying heating and cooling should be performed as rapidly as possible so that we can get the best quality of the product, which is depending on the nature of the product also. So it is in the statement 2, it is saying that for optimal quality, heating and cooling should be conducted slowly, whereas it should be rapidly. So this statement is incorrect. And statement 3 is also incorrect because since UHT processing, where we are heating any liquid for short uh, time, which is like few seconds, at the temperature above 135 degrees Celsius or 275 degree Fahrenheit, so that we can get the sterilized product. So it is of the order of seconds as per the definition of UHT, process treatment. So the residual time must be precisely controlled to avoid under processing. So it is say its processing time is not in minutes, it's in seconds. Whereas in this statement, it is saying that UHT process lasts for minutes. So this statement is also not correct. So we cannot say option B is correct. We will say option D is correct. 1 and 4 statement are correct. So have you understand this question? Is it clear? The person who has said option B.
Yes, ma'am. Yes.
So next question is, what is in the general the operating temperature range of UHT process of the milk?
Option C. 130 to
Option B.
170.
B.
Option C. Ma'am.
C. Option C.
Option B.
So
Option
we are getting two options, B and C, but C is correct. Why? Because
Ma'am,
in you, yes,
option A.
option A no, option C. So in UHT, what happens? We in the definition itself is saying that greater than 150 135 degree Celsius we should take, right?
Yes, ma'am.
So
But madam, this
it
is a time and temperature combination, na madam.
yes, it's time and temperature combination, but 121 is the autoclave. We know that it is the autoclave or the sterilization temperature, right? And if we see
Yes,
100
ma'am.
to 121, it is high heat treatment. Even it is greater than the pasteurization temperature, but it is not like it's it 121 is the sterilization temperature, but some products can be heated till for high temperature from 100 to 121, but it is even above the pasteurization. 121 is the sterilization, but this range is correct since it is greater than 135 degree Celsius. So this option is correct. Usually, what like in the industry is mostly 135 to 145, 150 or 150 temperature we usually use, either 145 or 150, 155 until this UHT temperature can be used. So here, like this UHT processing, this is also used for not for just milk, for the beverages like the fruit juices, for honey, wine, cream, soya milk, yogurt, soups, stews. So it can be used for various other applications, but mostly we process milk using UHT, because in the UHT there is the development of the burnt flavor. As we know in the UHT processed milk, so there is also disadvantage of this method. As we can, like, there was one research paper, uh, in that they used different food products and they treated with different UHT temperatures. So this is a paper, so they are doing research over it. So they have treated like the mango nectar, So juices, different juices like watermelon, carrot juice. So these can be these all these food products can be treated with UHT processing for sterilization purpose or getting the aseptic product.
During pasteurization also, ma'am, this temperature is maintained, right? 135 or something.
135 for pasteurization. I don't think usually the pasteurization is a low temperature process that is below 100 degrees Celsius.
Yes, yes. 60 to 75.
That is for milk. 60 to 70 is for milk. Like we have HTST and LTST.
Yes.
L,
Yes, yes.
LTL, yeah, low temperature for long time and high temperature for short time. But for that is for milk specifically you are telling. But there are other products like juices, jams, and they are also being pasteurized. But for them the temperature may vary, but it will be less than 100
Yes, yes.
if you are
80 to
doing
90 for juice.
pasteurization. Yes,
UHT,
so UHT is ultra high temperature processing, which is more than sterilization. We do sterilization as at 121, and it is even beyond that temperature, which is like around greater than 130 to 135. So we do it, and like we have the different companies milk like in the Tetra Pak kind of packaging, and that small packet is there. You can use it at the room temperature for even 6 months if you do not open the packet. If you open the packet, the aseptic condition will go and it may spoil. But if you until you are not opening the packet, it will be there at room temperature, that UHT process milk, for around 6 months. So which is already available in the market. So can we move to the next question?
yes,
Yes,
ma'am.
madam.
So this is our figure of Tetra Pak. So the figure provides illustration of the seals and the closures in the aseptic packaging. So we have 1, 2, 3, 4 as the
Option
different
A.
parts. We need to
B.
tell which
Option A.
which
Option A, ma'am.
option A. Then I heard C,
Option A.
option
Option
B,
Option A.
Option A.
A is correct, ma'am.
D.
many people are saying A.
D is correct.
D. So
Option A, ma'am.
Option
yes,
A.
option A is actually correct. So this one is the seal which is above or in this direction is like this. Oh, sorry. So we can see in this diagram these type of seals. These are known as the transversal seal, and the seals which are like vertical in the Tetra Pak kind of packaging material. So it is longitudinal seal. And if we open the packet and see this part inside this longitudinal seal, if we see there will be one strip like this, one strip is like this, and LS strip will be there. Longitudinal strip will be like this. So this strip, which is like there are two strips. So one strip is known as a strip side and another is a non-strip side. So non-strip side is at the bottom, strip side will be at the top, and these two will be connected using the longitudinal strip like this. This part is inside and this is outside. So inside will be our strip side and outside will be our non-strip side, and this will be connected with LS strip, which is longitudinal strip. So longitudinal seals we have, which is longitude like this. I'm talking about this one now. So it is a flat web of packaging material, and it is like sometimes it is formed like a tube. If you see this, it makes if you seal the whole one side of the packaging material like this, it will be like cylindrical kind of, or it will be like tube kind of. If you seal this side, it will be tubular like that. So a flat web of packaging material is formed into a tube which is sealed longitudinally, resulting in a cylindrical shaped structure. The strength of this longitudinal seam is determined partially by an overlap seal. So this is the overlap seal here. So its strength depends on the overlap seal, and partially by a plastic longitudinal strip. So when we seal, we have different layers. So when it is being sealed, like in these pictures, so we have LDPE, SDPE like this packaging materials, and so it also depends on how the plastic strip is attaching, or it is how much it is how well it is sealed. This strip is first sealed to one edge of the packaging material web, and once the packaging material tube has been formed, the to seal to the inner surface of the packaging material. So both these operations, the strip application and the actual longitudinal seal, are done by using sterile hot air or pressure. And there are different tests to check. This is the longitudinal seal. So there are different kind of materials we use. Also, like these seals can be tested using the conductivity test, reading test. Like in reading test, what usually is done, red ink is being put on the seal, and after like 5 minutes, all the red ink will be taken out, and if there is some kind of leakage in the seal, or the seal if it's not properly uh, like sealed, the longitudinal seal is not properly formed, then there will be this red ink will go in that part, and when you open, you can see there will be the spots of red ink like this. Transverse seal is, this is the transverse joints. So all these, like here, so it is done below the level of the product in the packaging material tube by constantly moving sealing and pressure jaws. Pressure is applied from the outside of the packaging material tube, squeezing the product from the sealing area. An electric impulse is passed through the sealing jaw, and heat is also transferred from the outside to the inside of the plastic layer of the packaging material. The polyethylene layer is heated, melted, and then pressed together between a pair of jaws. So it is being done under pressure. So pressure is also maintained so that the two layers can be stick together. And this type of seal can be they can be tested using leak test, peel test. So what happens uh, if the product is leaking from that side or that part, that means that the seal is not properly formed. Also the peel test is like there is one mechanical system. Here is also like one mechanical system, and here is the seal. So it we pulled or peeled, so we can see how much strength we need to pull that seal. So is it clear, this different type of seals? So this is
Yes, ma'am.
option A is correct, right?
Yes, ma'am.
Next question is pair the sterilization method used in the column 1. So column 1 is having different sterilization methods like system employing the moist heat, sterilization by the dry heat, and sterilization of the air by the incineration. And column 2 is telling about different temperature ranges. So we need to tell for moist heat and dry heat, and for air by incineration which temperature ranges are required.
Option A, ma'am.
Option B, ma'am.
So we are getting option A.
Yes, ma'am.
So option A is correct, that the system employs moist heat, which is the like for the sterilization. It uh, has a temperature of around 121 to 129 degrees Celsius, like we see in the autoclave, where we sterilize medical equipments and the laboratory equipments at 121 to 129 degrees Celsius. Sterilization by dry heat, its temperature is around 176 to 32 degrees Celsius, where we put we can maintain this kind of condition in the hot air oven or any kind of oven, where the glasswares they are used to sterilize sometimes oil, powders and metals. They are sterilized by this method at high temperature. And sterilization of the air by the incineration method, it is conducted at the temperature 260 degrees Celsius to 315 degrees Celsius. And these sterilized airs, they can be further, after the sterilization of the air, they can go in the fermenters, where we grow the microorganism. So it needs the sterilized air to be sent in the fermenter, so there will be no wild growth in the fermenter and pure air can go. So we have the option A, which is correctly guessed by you guys. So next question is, scrapped surface heat exchanger are utilized for only fluids that exhibit turbulent flow, fluids that exhibit laminar flow, and products that contain particles, fluids that exhibit turbulent flow, and products that contains particles. Or option D is fluids that exhibit laminar flow and products without particles.
Option B.
So We got option B. Any different opinion?
Ma'am, option C.
Option C. So option B is correct, not option C. It exhibits laminar flow, not the turbulent flow. So we know that there are three kinds of flows actually: laminar flow, transit flow, and turbulent flow. So laminar flows are the one which are smooth and streamlined and characterized by the water particles moving in the straight direct line within parallel layers, and it has the Reynolds number, low Reynolds number, whereas turbulent flow are irregular or chaotic, faster moving water characterized by erratical mm, moving water particles. They mix the parallel layers, they will get mixed. That mix between the parallel layers, and its Reynolds number is very high. So these are the basic that these are different flows. Now our question is on the scrap surface heat exchanger. So what happens in the scrap surface heat exchanger? It looks like this. So when there is this operation, when we operate it, the product is brought in contact with the heat transfer surface like this. If we see, here is our product and these are the scraps or the blades, and this part is the heat exchange surface, this part. So product will be in contact with the heat transfer surface that is rapidly and continuously scrapped. That's why we are called it scrapped surface heat exchanger, thereby exposing the surface to the untreated products. In addition to maintaining high and uniform heat exchange, the scraper blades, they provide a degree of simultaneous mixing and agitation also. So we uh, mix the product in the scrap surface heat exchanger. The product is mixed and agitated. Also, it is heated. So it is basically good for viscous, sticky uh, products which have the particulates and produce crystals. Also, scrap surface heat exchangers, they basically used for the laminar flow because it is being used for the high viscosity fluids. That's why we need scrap blades, scrappers or the blades. And since it is of high viscosity, only laminar kind of flow can be maintained, and it is basically used for the particles of the products having particles. So we have our option B as correct. It is used for laminar flow, not for the turbulent flow, and it is used for the products which contains particles and not for the products which does not contain particles. Is it correct or clear? Can we move to next
Clear.
question?
Yes, ma'am.
Yes, ma'am. Yes,
The
ma'am.
next question is a problem. So we need to solve this problem. An orange juice drink is aseptically packaged and kept at room temperature, which has 50 mg of vitamin C. And at the time of this 50 mg, of vitamin C is initial concentration. That means at the time of packaging, we have 50 mg of vitamin C in orange juice. During storage, the vitamin C is degraded and following the first order reaction with a degradation rate constant of 2.1 into 10 to the power minus 3 day inverse. Determine the remaining concentration of vitamin C after 30 days of storage. Please try to solve this. So it is saying that in the question that we need to apply first order kinetics equation. So we have the degradation rate constant K, which is 2.1 into 10 to the power minus 3 per day. We have the storage time duration as 30 days. The initial concentration of vitamin C is 50 mg and we need to calculate the final concentration. Since it is following first order kinetics, so we can use this formula C equals to C naught e to the power minus kT. So can we apply this formula for first order kinetics or first order reactions? Can we apply? I'm asking you guys.
Yes, ma'am.
Yes, right? We can apply. So we can write this as C by C naught, C by C naught equals to e to the power minus kT. So which can be written as ln C by C naught equals to minus kT. So we have this formula where ln C by C naught means final concentration divided by the initial concentration. So initial concentration is given in the question as 50 mg. So C by here we can write 50 equals to minus kT. K is rate constant, degradation rate constant K. So it is given as 2.1 into 10 to the power minus 3 per day. So minus 2.1 into 10 to the power minus 3 into storage time, which is 30 days. Since it is also in per day and storage time is also in days, so we can use days as unit of time here. So T equals to 30. ln C minus ln 50. This can be written as ln C by 50 is equals to ln C minus ln 50 equals to 2.1 into 30 is 63 into 10 to the power minus 3. Now ln C equals to, this can go on the left hand, on the right hand side. So ln 50 minus 63 into 10 to the power minus 3. So ln C is equals to ln 50 means 3.912 minus 63 into 10 to the power minus 3. So ln C equals to, after subtracting this, we will get 3.849. This is LNC value. So C is equals to e to the power 3.849, which is C equals to 46.95 mg. So option C is in our range. So option C is correct. Is it clear, this question to all?
Yes, ma'am.
Okay, thank you.
Yes, ma'am.
So let's move to the next question. It is also one numerical problem. So in aseptically packaged lemon juice, the starting concentration of ascorbic acid is 90 mg per 100 grams, which decreases to 70 mg per 100 grams after 150 days of storage. This loss adheres to first order kinetics, characterized by a rate constant as 0.00542 per day and an activation energy of 70,110 joule per mole at room temperature 25 degrees Celsius. Using the rate constant R equals to 8.314 joule per mole per Kelvin, calculate the storage temperature in degree Celsius. So we have here now lemon juice, and ascorbic acid concentration is degrading as like it is similar to the previous question. So we have the concentration given that from 90 mg to 70 mg per 100, the concentration is decreasing in 150 days, and we need to we have the activation energy also. So that means we need to correlate with the temperature, and we need to then find the storage temperature. So let's solve this question, that the degradation rate constant is given as 0.00542 per day. Storage time is 150 days. Initial concentration is 90 mg per 100 gram. Final concentration is 70 mg per 100 gram. Activation energy is 70,110 joule per mole, and temperature 1 is given as 25 degrees Celsius. So we can calculate in Kelvin, which is 25 plus 273 equals to 298 Kelvin. Since the degradation of vitamin C is following first order kinetics, so like we did in the previous question, we are applying this first order kinetics formula. Ln C by C naught means ln final concentration divided by initial concentration equals to minus K2 into T, which is the degradation rate constant into time. So we know that the initial concentration is given as 90 mg per 100 grams. So ln the initial concentration, which is at the denominator, is 90 mg per 100 gram, and the initial concentration, that is initial concentration, and the final concentration is 70 mg per 100 gram, which is at the numerator. So 70 by 90 equals to K is not known here, and the time interval is 150 storage days. So 150, right? T equals to 150. Now ln 70 by 90 can be written as ln 70 minus ln 90 equals to minus K2 into 150 storage days. So K2 is equals to ln 70 means 4.249, this value, minus ln 90 is 4.499 divided by 150. So it will be 4.249 minus 4.499 is equals to minus 0.25 divided by 150, which is 1.67 into 10 to the power minus 3 per day, and minus this is cancelled out with this minus K2. So K2 will be 1.67 into 10 to the power minus 3 per day. Now, since we need to calculate temperature, we need to apply Arrhenius equation here. So, ln, we know Arrhenius equation, right? So, ln K2, K1 by K2 equals to EA by R 1 upon T2 minus 1 upon T1. So, ln K1 means the first, constant at the first temperature by rate constant at the second temperature is equals to EA is the activation energy, R is the universal gas constant into 1 upon T2, which is the temperature in Kelvin at T2, which is not given to us, and T1 is the temperature, initial temperature in Kelvin. So ln K1 is given in the question as like here 0.00542 per day. So K1 is 0.00542 divided by K2, which we just calculated from here, which is 1.67 into 10 to the power minus 3 is equals to EA is given in the question as this 70,110 divided by R, which is a constant, so 8.314 bracket 1 upon T2, which is not known to us, we need to calculate this, minus 1 upon 25 degrees Celsius, which is 298 Kelvin. So from this we need to further just simplify. So ln 0.00542 divide by 1.67, so minus, so when we simplify it will be ln A minus ln B, right? So minus ln 1.67 into 10 to the power minus 3 equals to, this will be RHS will be same, 70,110 divided by 8.314 1 upon T2 by 1 upon T1, which is 298 Kelvin. ln 0.00542 is minus 5.22, then minus ln 1.67 into 10 to the power minus 3 is minus 6.40 equals to 70,110, this term, by 8.314 we divide, so we can get 8432.76 and multiply 1 upon T2 minus 1 upon 298. So this term we can keep one side and others we can transfer. So 1 upon T2 minus 1 upon 298 equals to this term. If we solve, it will be minus 5.22 minus minus plus 6.40, which is 1.18. Divide by this term 8432.76. So after divide we will get 0.00014. This is the value of 1 upon T2 minus 1 upon 298. So we need to transfer this on the right side. So 1 upon T2 equals to, this is minus, so it will go and it will be plus. So 1 upon 298 will be 0.00336. So plus 0.00336 plus 0.00014. So it is 0.0035. So 1 upon T2 value is this. So T2 value is 1 upon this term 0.0035. So it will be 285.71 Kelvin or minus 273, which is 12.71 degree Celsius. So our option B is correct since it is in this range. Is it clear to everyone?
Okay, ma'am.
Yes, ma'am.
Has anybody solved this with me? Is a calculation correct? Okay, so as you said, it's clear to everyone. We are moving to next question, which is on hurdle technology. Which one of the following is not a fundamental principle or goal of hurdle technology? A. Directly eliminating microbial spores. B. Influencing microbial stability through the interplay of various factors. C. Inhibiting the proliferation of microorganisms. D. Disrupting the homeostasis of microorganisms. So we know in hurdle technologies, we apply several technologies together and at different intensities. Like we apply temperature, we either we use low temperature or the high temperature or low water activity sometimes. pH we can change, alkalinity to acidity, redox potential sometimes we change, and we can apply high pressure. So all these treatments we do together and at different intensities. So microorganisms, if it can like cross all these barriers, like this barrier, if it is able to cross, so it's all the cellular energy will go until it will reach to final process and it will not able to cross. And it by this method we can get sterilization of the product and at the low intensity treatments which cannot harm our product. So anyone, not a fundamental principle of hurdle
Option
technology.
A.
Option A.
Ma'am. A, ma'am.
Option A.
Option A, ma'am.
Yes, all of you are correct. Since we know in the hurdle technology, we use different preservation factors or the hurdles that disturbs the homeostasis of microorganisms. And microorganisms, it will not jump over all the hurdles which are present in the food. So the preservative factors, they prevent the microorganisms from multiplying and causing them to remain inactivate or even die. We do different series of the treatments. The hurdle concept illustrates that complex interaction of temperature, water activity, pH, etc. to get the microbial stability in the food. And this direct killing of the spores, it is the method for sterilization. It is done by sterilization, not by hurdle technology. Like it can, but it may not, like may be possible or may not be possible. So we are moving to the next question now. The method that utilizes a mix of pressure, heat, and ultrasound for the inactivation of microorganism is known as nanothermosonication, ultrathermosonication, manothermosonication, or control thermosonication.
Option C. C, ma'am.
Manothermosonication.
Yes, ma'am. Option C, ma'am.
Yes. So manothermosonication is the method where there is mix of pressure, heat, ultrasound, all. Mainly this term, these all terms, they are not very much uh, like they are not uh, used, and the thermosonication, this word itself, it means that we are combining the sonication and the heat. Sonication means like the sound waves, and moderate heat we apply so that we can get the greater effect of the inactivation of the microorganism as compared to only alone the sonication or alone heat. In manothermosonication, there is heat, ultrasound, and pressure, as we just now discussed that our option C is correct. So here we apply temperature, pressure, and because of that, like when we are applying, because of the pressure and the temperature, the sonication effect, which is the cavitation and forming the, like basically forming the bubbles, and the bubble will grow and it explodes. So ultrasound is based on this method. So cavitation, like the efficacy of cavitation or the ultrasound, it increases and there is more inactivation of the microorganisms. So microorganisms that have high thermotolerance can be inactivated by manothermosonication, because here we are applying three treatments together. And this type of system is like pilot on the pilot scale or on the lab scale is used for thermosonication. Like here in this double jacketed chamber, sample will be there. So from nitrogen gas we are applying pressure. Here it is ultrasonication, and because of this is temperature, from this temperature is coming. So all are affecting the product, or all are treating in the like the product in the this double jacketed chamber, and from here we can take out our product after treatment. So far is clear for everyone? No doubt so far?
Yes, ma'am.
Yes, ma'am.
Okay. So let's see this question. Column 1 is having different food products like tomato, milk, apple, and column 2 is having the pH. We need to tell which product is having, like lies in which pH range.
Option C, ma'am.
Option C, madam. Option C.
Option C, ma'am.
First, second
Option B.
means you are saying
Option B.
option B.
Option
One
C, ma'am.
C, ma'am. C.
maximum are saying C. Okay, so we got two B's.
Option C, ma'am.
Option C is correct actually. The apple is basically having the so very low pH, less than 4, which is due to the presence of malic acid, which is an organic acid present in apple. So it's in apple the pH is around 3.3 to 3.9. In tomato, the pH is around 4.3 to 4.9, and it has oxalic acid in this. Because of that, it is coming under low acid food. So we can see in the pH scale, apple lies here. Strongest, which is at the 0 pH, is the strong acids like HCl, H2SO4, the concentrated acids. Then lemon is the like it's the natural food which is having more which is having low pH or more acidic than apple. Then here is a tomato. Water is something like near neutral. Blood, it's around like above 7 to 8, and other products. Milk is basically, it lies in the range of 6.7 to 6.9, here near neutral, because like it also have one organic acid in this, which is lactic acid. Actually, lactose is being converted in the lactic acid by the lactobacillus species. So this is the different pH ranges. Is this clear? Option B is not correct. Tomato is less acidic than apple. So can we move to next question?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
So in column 1, we have different packaging materials like antimicrobial packaging system, LDPE, then EVOH, which is ethylene vinyl uh, alcohol, and then we have the fourth option as composite LDPE, composite of LDPE and ethylene vinyl alcohol. And option column 2 is telling about different hurdles like the moisture barrier, oxygen barrier, combined moisture and oxygen barrier, combined moisture, oxygen and microbial barrier. So these packaging materials also provide some kind of like barrier and or the hurdle, so that microorganisms cannot attack on the food product.
Option A, madam.
Option B, ma'am.
So we have two options, A and B. Sorry, option A and B. So option B is actually correct. Why? Because in single layer packaging, such as the LDPE, ethylene vinyl alcohol, moisture and oxygen act as the sole barrier respectively. So EVOH and LDPE So LDPE provides barrier against the moisture, and EVOH provides barrier against oxygen. Then we have composite and antimicrobial packaging system. So in an antimicrobial packaging system, we have microbial barrier functions. And in so option first goes with fourth. We have moisture barrier, and packaging system can means anything which is LDPE or EVOH. So it can provide moisture and oxygen barrier also. And the composite of LDPE and EVOH. So since one is providing one property, like one barrier property, so their composite layer will provide jointly barrier against the moisture and oxygen. So we have option B as correct.
Okay.
So is it, yeah, so can we move to next question? So option A is not correct, B is correct.
Okay.
So, okay. Next question is pathogens cannot multiply and spores fail to germinate at a pH of greater than 4.5, less than 4.5, 5.5, 6.5. In this pH, even pathogen cannot they grow, and spores also cannot germinate.
Option B.
Option B.
Option B, ma'am. B.
Less than 4.5.
Yes, so it is highly acidic condition. In this, pathogens and the spores also cannot survive. So we can find this kind of uh, pH system in different fruit juices or the sauerkraut, which is a fermented product of the cabbage. So because of the fermentation, uh, pH goes low and pathogens and spores cannot survive. So next question is column A is having different antimicrobial agents as acetic acid, propionic acid, sorbic acid and lactic acid. In column B, we have different like their origins, which acid is found in which kind of food product, which is yogurt, rowan berries, which is also known as ravan berries, but we can pronounce it rowan berries, and vinegar, Swiss cheese.
Option B.
B, ma'am.
Option B.
Option B. B, ma'am.
Yes.
B.
Option B, madam.
Yes.
B.
Option B. Yes, everyone is correct. So vinegar is a source of acetic acid, right? Propionic acid is basically produced in Swiss cheese. Also, like the bacteria which produces uh, propionic acid is the Propionibacterium. So this particular species is also used. It just does not provides propionic acid. It also produces CO2 gas in the Swiss cheese because like when it goes under the fermentation, so CO2 gas is produced, and because of that the holes, or which we known as the eyes, is produced on the cheese, Swiss cheese. Sorbic acid is available in the rowan berries, and lactic acid is produced by lactic acid bacteria, and usually it found in the dairy products like curd, yogurts, etc. So, so what happens? These all are organic acids, and it lowers the pH in a food product. So when the pH is low, microorganisms cannot grow. So it is a natural defense system of the fruits or different food products, because at the low, what is happening, these acetic acid, propionic acid, sorbic acid, lactic acid, they passively diffuse in the undissociated form. So as such, like the acetic acid as such will go in the cell wall, like it is hydrophobic, so it will bind to lipids, in which is the lipid bilayers in present in the cell wall. So cell lipids which are present in the cell wall, it will going to bind with that, and it can this acetic acid or the organic acid, it can pass through the cell wall and can go inside the bacterial cells, where it will lower the intracellular pH, and it can affect the affect different different functions or the enzymes. It can degrade their in the enzymes are basically the proteins. So in the low pH, the proteins may get denatured. So like this, if the enzymes structure is denatured, so it will not able to do the regular function. So it can affect the cell walls regular, so the bacterial cells regular function of survival. Also the low pH, like it can overall affect the homeostasis of the bacterial cell. So these organic acids, they contribute to the, like they lower the pH, and thereby the microorganisms cannot grow.
Okay, ma'am.
Yes, so next question is, the process that involves initial partial drying in a concentrated solution and then freezing is referred to as dehydrofreezing, osmodehydroconcentration, osmodehydrofreezing, or concentration freezing. First, we are
Option C.
partially drying in a concentrated solution and then we are freezing. So
Option
option
C, ma'am.
C.
Option C.
So everyone is agreeing to option C. What is
Yes, ma'am.
dehydrofreezing? In this, fruits and vegetables, they are preserved using evaporation of the of 50 to 60% of water before freezing. So first we dehydrate and then we freeze. The texture and the flavor are superior than those of the dehydration and freezing alone, and rehydration is more rapid than with the dehydrated product. So it is better than dehydrated or freezed product. Second is osmodehydroconcentration. So usually what is happening here is, this is concentration, this is not freezing. So here in the question we are asking about freezing. So option B is not correct. Instead, option C is correct. Then is concentration freezing. Concentration freezing is the technique where concentrated liquid foods, they are pre-freezing step. They goes under pre-freezing step. After that, there is a separation of the pure ice crystals. Once crystallized, the water fraction is removed, and the total soluble solid content will be higher in the solution. So what we are just freezing the water into ice, and the ice is being removed. So concentrated product we get. So it is known as concentration freezing. Osmodehydrofreezing, it's basically a combination where a limited air dehydration step is preceded by osmotic treatment in the sugar solution to obtain fruit products at low water activity and to be stored at low temperature. And these products, they are basically free of preservatives and more similar to fresh like food. Their flavors, colors and textures are mostly the same, and the lesser quality of the food product is affected, so they are used. So osmodehydrofreezing has been proposed for the preparation of fruit ingredients at reduced moisture with functional properties suitable for different applications. So we have option C as correct. So we are moving to next question, which is we need to identify the enzyme which is responsible for catalyzing hydrolysis of beta 1,4 glucoside bond between N-acetyl muramic acid and N-acetyl glucosamine in bacterial cells. Option A is N-acetyl muramide glycan hydrolase. Second is lysozymes, and third is trypsin. So only first one is correct. First and two, or only three. What is the attendance today? 39 students. Anyone?
Option 1 and 3, not sure, ma'am.
C.
It's
C, ma'am.
option B.
Option C, ma'am.
Option C.
C.
C is correct. 1 and 2. Basically, lysozyme, which is also known as N-acetyl muramide glycan hydrolase. So these two enzymes are the same. First and two are same, which is the same enzyme and doing the act, like it is catalyzing the hydrolysis of beta 1,4 glucosidic bond between N-acetyl muramic acid, which is we known as NAM, and N-glucosamine NAG of the bacterial cell walls, and these are found in egg, milk, tears and other secretions, and insects and fishes, and in hypotonic solutions. And the enzymes causes the and these are the causing the lysis of the bacterial cells. So we know that we in our tears we have lysozymes enzyme, so it can lysis the bacterial cell wall. So it is a natural antimicrobial agent which is present. So this is the structure of like this. Where is the cursor? So this is NAG, NAM. So like this, they are connected. Here is the site where lysozyme cleavage happens. So this bond, it is going to cleavage. And since we here it is written as that these are found in hypotonic solutions. So from the picture, can anyone tell what are these different solutions of the definition of these solutions based on, we classify based on the osmolarity or osmotic pressure, like hypotonic solution, isotonic solutions, and hypotonic solutions. We have picture also. So please help me.
If it is a hypertonic solution, that means the solute concentration is more rather than the solvent concentration for hypertonic solution. For example, if we'll place a cell in a hypertonic solution, their
Hmm.
exosmosis will occur.
Hmm.
For isotonic solution, there will be like the concentration inside the cell and the surrounding is equal. So there will be no migration of any solvent molecules.
Hmm.
And in hypotonic, the solute concentration is less as compared to the solvent concentration. So there will be
In
endosmosis.
outside, right?
Yes, ma'am.
Yes, yes. Very nice. She has explained very nice. So cell in this case swells, right?
Yes, ma'am. Like when we are placing a cell inside these three kinds of solution, whether exosmosis will occur or endosmosis will occur, depending upon the relative concentration in which the cell is placed.
Yes, yes, very correct. Very nice explanation. So let's move to the next question, which is dash is a carcinogenic substance that forms from nitrite and commonly used in the preservation of the meat and poultry products. So we have these four as our options. So out of these, which is a carcinogenic substance which is formed from
And
nitrites? Which option?
Option C is not sure whether, madam. Option C.
option C,
Nitrosodimethylamine.
and nitrosamine formation, I think nitrosamine.
Mhm. You are guys correct. So nitrates and nitrites, they are basically added to the preserved meats as a preservating agents, and they are usual, like they are carcinogenic substances, and they form the carcinogenic substances, and that's why nowadays there is a debate that which kind of food additives should we add in food products so that in during the processing they don't convert into the carcinogenic substances and do not cause any toxicity to humans. So these nitrates and nitrites, they are associated with a high risk of gastric cancers. Nitrosamines, they are carcinogenics and they involves in the chemical reactions with various other nitrates, nitrites, and various proteins. And NDMA, which is dimethylamine, it it can be encountered to us in our diet. So this is the nitrite ion. It goes like it takes H plus and it becomes nitrous acid, and it another H and H2O will going to remove, and this ion will form nitrosonium, and then further this an ion, they it can interact or react with dimethylamine and form NDMA, which is a carcinogenic substance in the meat and poultry products. So you were correct. Now there is a question on food lipids, oils. So among the oils listed below, which has the greatest concentration of lauric acid? Mustard oil, sunflower oil, peanut oil, or the palm oil? Lauric acid. So we know that the fats and the fatty acids, they can be classified as the saturated fats. Like saturated are mainly animal fats, butter, lard, and the unsaturated fats, which are mainly oils. So unsaturated fats, they can be PUFA, which is polyunsaturated fatty acids, and the MUFA, which is monounsaturated fatty acids. So in polyunsaturated fatty acids, omega-3 fatty acids and omega-6 fatty acids, we have these two categories. So in omega-3 we can have EPA, DHA, ALA, and it is mostly like in the either in the nuts or in the fish or the shellfishes. Omega-6 fatty acids, it is found in the corn oil, safflower oil, sunflower oil. Omega-6 is omega-9 is in the olive oils, avocado, peanuts, and almonds. So PUFA and MUFA, we have also saturated fats. So like we have linseed also in that omega-3, which is very helpful for DHA, is very helpful for growth. uh, Omega-6 is can be linoleic acid. It can be found in different plant sources like sunflower or the safflower oil, in all these oils. Omega-6 is popularly, we know, oleic acid. So these can also be nuts, avocados, etc. Saturated fats like palmitic acid, it can be in egg, dairy products, coconut oil, red meat, etc. So now we have the
Palm,
question among the oils. Yes,
option D, ma'am.
option D. So we have question about these are the oils and we have greatest concentration of lauric acid. So what is like in out of which category lauric acid falls? Please, can anyone these categories? Is it a
Saturated fats.
saturated fats? Lauric acid.
Oh,
So and it is in oils. So oils, see, oils, they are basically in unsaturated mostly. But yeah, lauric acid is basically a saturated fatty acids. So it is a 12 chain carbon structure like this. So it is found in palm kernel, coconut oils. Classified in this, the high amount of lauric acid has been found. Usually oils doesn't have lauric acid, it has low lauric acid because it is a saturated fatty acid, and oils, they comes under unsaturated, right? But these oils, palm kernel oil and the coconut oil, they have high concentration of lauric acid. This is unique as no other plant oil contains more than 1% lauric acid. In mustard oil, we have mostly uric acid, which is MUFA, one kind of MUFA. And in sunflower oil, we have linoleic acid and oleic acid in the high amount. In peanut oils, we have oleic and linoleic acid in the high amounts. So, you were correct. Palm oil is the correct answer. So, let's move to next question. Which among the below given statements are incorrect regarding triglycerides from animal sources? So, A is they are more susceptible to hydrolytic rancidity than oxidative rancidity. Option B is they are generally a soft solid at room temperature. C is they contain a high amount of unsaturated fatty acid and seldom experience hydrolytic rancidity. And D is they have a high RM number. So first tell me what is RM number? Anyone? We have different numbers regarding the lipids like saponification number, or we have iodine value, Polanski value, RM value, right?
Yes, ma'am.
So anyone please share some of the views regarding that. What is these different? I am not
Uh,
audible.
I- yes, ma'am.
I'm audible, right? I
Yes, ma'am.
get a message. Not audible.
Yes, ma'am. Okay, ma'am. Now audible, madam. Audible, madam.
Okay, thank you for confirming. So let's discuss what are different these values. Anyone please can
Yes,
share?
ma'am. The iodine number says about the degree of unsaturation.
Hmm.
Uh, Likewise, the saponification value is what, like, the amount of potassium or sodium hydroxide which is used to neutralize the fatty acids. And when it comes to RM value, it is the measure of the amount of short chain fatty acids which is present inside a fat or oil.
Okay. What is difference between RM and Polanski value? Like it's very similar, no? So she has told two of
Against
the
it's volatile fatty acids, right?
Polanski, yes, Polanski value.
Yeah, like it measures the amount of volatile fatty acids which is present in
Hmm.
the oil.
Then RM value, it how it like they how what it measures and what Polanski doesn't like that. They are like kind of opposite.
Then don't have much.
Amount used to neutralize the soluble volatile fatty acid.
Hmm. So let's discuss this first and then we will solve this question. So we have iodine number, as she rightly mentioned, it measures the degree of unsaturation of an oil, fat or wax. This number indicates the mass of iodine, which is this I2, in grams, how much amount that is necessary to complete saturation in this unsaturated molecule or unsaturated molecules, the molecules of 100 gram of the given oil. So in 100 grams, how much iodine is required to uh, make this unsaturated uh, oil into the saturated one. So this bond will break and I will go and attach here and here, and it becomes saturated. So this is iodine number. Now is saponification number, which represents the number of milligrams of the either the potassium hydroxide or sodium hydroxide requires to saponify 1 gram of fat under the conditions specified. Like we have this kind of fat, either KOH will go or NOH we can use, and this bond and this bond will going to break, OH will go and attach, and this and this will separate out as these entities. So this is what happening actually. So we will calculate 1 gram of, like to saponify 1 gram of the fat, how much KOH is required. So that is saponification number. So it is a measure of the average molecular weight or chain length of all the fatty acid present in the sample in form of the triglycerides. The higher the saponification value, that means lower the fatty acid average length. The chain length will be lower, inversely related. The lighter the mean value, lighter the mean value of molecular weight of triglycerides, like if the higher saponification value means lower the chain length, or we can even say in another terms, in molecular weight terms, that the lower is the molecular weight. So it is inversely proportional to chain length and molecular weight. Next is RM value. It is the number of ml of 0.1 NaOH solution required to neutralize the steam volatiles, water soluble fatty acids distilled from oil or fat under precise specific condition. So it measures actually water soluble steam volatiles fatty acids like the butyric or the caproic acids which are present in the oil. Polanski value, it is also the number of ml of 0.1 NaOH solution which is required to neutralize the steam volatile water insoluble fatty acids. So this is the difference. This is the uh, like the minimal. This is the these two values are calculated in the same way, but one is calculating water soluble fraction, and the another is calculating water insoluble fraction. So the insoluble fatty acids distilled from oil or fat under precise specific conditions. PV measures is a measure of steam volatile and water insoluble fatty acids like caprylic acid, capric acid or lauric acid which are present in the oil. And okay, we calculate the formula, we calculate RM value and Polanski value. So A minus B into N into 11, which is volume of NaOH solution required for the test minus for the blank into its normality of NaOH into 11, and Polanski value is 10 into the volume which is required into normality. So this is the base. One is calculating the water insoluble part of the fatty acid and one is water soluble. So is it clear? This is the
Yes, ma'am.
solution of our yeah. So this is the solution of our problem, which is which among the below given statements is incorrect regarding the triglycerides from animal sources. So incorrect is C. This is not correct. They contain a high amount of unsaturated fatty acids and seldom experience hydrolytic rancidity. So what happens in animal fats? Triglycerides are generally more prone to hydrolytic rancidity, which leads to the production of free fatty acids and relatively rich in saturated fatty acids. Solid at the room temperature and less iodine number and higher RM value. Whereas the plant triglycerides, they are rich in unsaturated fatty acids, so they are liquid at room temperature and high iodine value and lower RM value. So, uh, so from this we get to know that animal fats, they are rich in saturated fatty acids, whereas plant triglycerides are rich in unsaturated fatty acids. And since option C is not correct because it is saying that they contain high amount of unsaturated fatty acid. So can we move to next question?
Yes, ma'am.
Yes.
So column 1 is having different vitamins, vitamin A, D3, E, K1, K2, and column 2 the chemical name tocopherol, menaquinone, phylloquinone, and retinol and cholecalciferol. So these are, we know that there are no commonly vitamins, vitamin A, D, E, K, like that, but we do not generally pronounce their chemical name.
Option C, ma'am.
Option C. C, ma'am.
C. Option C.
So everyone is correct that vitamin A
Yes, ma'am.
is known as retinol. Vitamin D3 is known as cholecalciferol, right? And then vitamin E is
Tocopherol.
tocopherol, right? Fourth is, which is K1. Vitamin K1 is
Phylloquinone.
phylloquinone. And fifth is K2 is menaquinone. So everyone knows. So this is the end for today's session. We discussed today about aseptic processing and packaging. Then we discussed about the commercial sterilization and aseptic processing and their application in the food industry. Then hurdle technology concepts. Then we discussed about natural antimicrobials like lysosomes and their mechanism of action. And then we discussed about different fatty acids or the fats. And in the next class, we will discuss about how the oils are being extracted, they'll be refined, and how and different their modification, like how are the different modified fats. So after this, I'll be uploading this uh, recorded video on my YouTube channel and PDF on the Google Drive. And if you have asked with any doubt, you can ask now, please. And if you don't have any doubt, you can leave the meeting. Thank you so much for joining today.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, everyone, for joining. Let's see next week.
Thank you, ma'am.
Let's meet on the next week.
Thank you,
Thank you so much.
Thank you, ma'am. Thank you, ma'am.
Thank you so much for joining.